Hello everyone. In this class, we'll try to understand how oxygen is transported in the blood. Modes of oxygen transport. Oxygen is transported from lung to the tissue in two methods. One is physically dissolved in the plasma and in combination with hemoglobin in the red cell. Approximately 98% of the oxygen is transported in combination with hemoglobin and the remaining 2% in the physically dissolved form. In physically dissolved form, in plasma, only about 2% of oxygen is transported. The amount of physically dissolved oxygen in the blood can be predicted by Henry's law. Henry's law states that amount of gas that dissolves in a liquid at a given temperature is proportional to the partial pressures of the gas. Thus, the quantity of dissolved oxygen in arterial blood is calculated from the following equation. That is, dissolved oxygen is equal to oxygen solubility into partial pressure of oxygen in the arterial blood. So that is equal to 0.003 into 95 mm of Hg, that is normal PaO2, that equals to 0.3 ml per 100 ml of blood. Thus, PO2 of 95 mmHg, the dissolved oxygen is 0.3 ml per deciliter. That means in a normal healthy adult, about 0.3 ml of oxygen is transported in dissolved form in 100 ml of blood. As the cardiac output is 5 liters per minute, at rest oxygen transported in dissolved form at rest is about 15 ml per minute. Therefore, oxygen transported in dissolved form alone is grossly inadequate to meet the oxygen requirement of the body, which is about 250 ml per minute at rest. More than 98% of oxygen is transported in combination with hemoglobin. This becomes possible due to the binding capacity or binding affinity of hemoglobin for oxygen. Hemoglobin that binds with oxygen is called as oxyhemoglobin, and hemoglobin that does not bind with oxygen is called as deoxyhemoglobin or reduced hemoglobin. Formation of oxyhemoglobin. The hemoglobin molecule is a protein made up of four subunits that are bound uh, together. Each subunit consists of molecule group known as heme and a polypeptide attached to the heme. The four polypeptide of a hemoglobin molecule are combinedly known as globin. Each group contains one atom, that is iron, which is in ferrous form, which binds with the oxygen. This is a structure of molecule. You can see the four heme groups, okay, so which are attached to the iron in the center, which is attached to the globin. Since each iron atom can bind to one molecule of oxygen, a single hemoglobin molecule can bind four molecules of oxygen. The hemoglobin binds with oxygen only when iron is in ferrous state. The ferrous iron in hemoglobin is oxidized to ferric iron to form methemoglobin. Thus, methemoglobin can't bind oxygen. Methemoglobin formation occurs under influence of various compounds like nitrates and sulfonamides. Methemoglobin is also formed spontaneously. However, the enzyme hemoglobin reductase is present in red cells that reduces the methemoglobin to hemoglobin. Therefore, normally only 1.5% of the total hemoglobin is methemoglobin. Deficiency of hemoglobin reductase, a genetic defect, increases the methemoglobin concentration and decreases the oxygen carrying capacity. Oxygen binds rapidly and reversibly to hemoglobin to form oxyhemoglobin. The R and T states of hemoglobin, that is relaxed and tense states of hemoglobin. During this process of HbO2 formation, the heme remains in ferrous state. Thus, this process is oxygenation and not oxidation. As there are four subunits in hemoglobin molecule, the hemoglobin reacts rapidly with four molecules of oxygen to form oxyhemoglobin, that is HbO8. When oxygen is not bound with hemoglobin, that is deoxyhemoglobin, the four subunits of hemoglobin are tightly bound with each other. This configuration of hemoglobin is called as tense or T state. In this T configuration, affinity of hemoglobin for oxygen is less. When first oxygen molecule binds with hemoglobin, the four subunits enter into the relaxed state that exposes the more oxygen binding sites. Therefore, in our configuration, the affinity of hemoglobin for oxygen is greatly increased as many as 200 to 500 times. When PO2 is very less, the most of the hemoglobin molecules are in T state, that is 10 state, and have a low oxygen affinity. When PO2 is very high, the hemoglobin molecules are in R state and they have a high oxygen affinity. Thus, the quantity of oxyhemoglobin formation is the function of partial pressure of oxygen in the blood. In pulmonary capillaries where PO2 is high, the reaction favors to form more oxyhemoglobin, and in tissue capillaries where PO2 is low, the reaction favors formation of deoxyhemoglobin that helps oxygen to be unloaded from hemoglobin and becomes available to the cells. So this is a T or a taut formation, taut conformation or tensed uh, conformation to the relaxed state, that is R conformation. Okay, so you can see when oxygen binds, uh, the salt bridges are broken down and the hemoglobin becomes uh, gradually more develops more affinity towards the oxygen. Okay, and this is the tense formed or T form or taut form, and this hemoglobin is a relaxed form. Each gram of hemoglobin binds with 1.34 ml of oxygen. The maximum amount of oxygen that can be carried by hemoglobin is called the oxygen carrying capacity. In a healthy individual, the oxygen carrying capacity of arterial blood is about 20 ml of oxygen per 100 ml of blood. Considering the hemoglobin concentration is 15 grams, so that equals to 1.34 ml into 15 grams, that comes around 20.1 ml of oxygen per deciliter of blood. The oxygen content of hemoglobin is the amount of oxygen actually bound to hemoglobin, whereas oxygen capacity of hemoglobin is the amount of oxygen that can potentially bind with hemoglobin. The percentage of saturation of hemoglobin with oxygen is the ratio of the quantity of oxygen actually bound, that is oxygen content, to the quantity that can be potentially bound, that is oxygen capacity, multiplied by 100. Oxygen hemoglobin dissociation curve. The oxygen hemoglobin dissociation or equilibrium curve or dissociation or association curve explains the relationship between partial pressure of oxygen in the blood with the oxygen saturation of hemoglobin. The oxyhemoglobin equilibrium curve is an S-shaped curve over the range of PO2 from 0 to 100 mm of Hg. The sigmoid, the sigmoid shape of the curve results from the hemoglobin affinity for oxygen at various partial pressure of oxygen levels. As PO2 rises, the hemoglobin saturation uh, progressively increases. However, the saturation is not linear with increase in PO2, for which the curve becomes sigmoid shaped. The S-shaped oxyhemoglobin equilibrium curve enables the oxygen to saturate hemoglobin under high partial pressures in the lungs and to give up large amounts of oxygen with the small changes in the PO2 at the tissue level. So this is the association of the the partial pressure of oxygen to uh, the percentage of saturation of hemoglobin. Okay, so as the partial pressure of hemoglobin increases, the saturation is also going to increases. In the initial phase, it rapidly increases, and later at around 60 to 70, it becomes plateaued. Okay, and this association, I mean, which is a sigmoid shape, I mean, it is called as oxygen dissociation curve. This curve is divided into two major phases, the steep phase and the plateau phase. The steep phase, this curve has a steep between a PO2 of 10 to 60 mm of Hg. During this phase of the curve, combination of oxygen with hemoglobin increases very rapidly as the PO2 increases from 10 to 60 mm of Hg. The oxygen saturation of hemoglobin is about 90% at PO2 of 60 mm of Hg. So, what is the significance of this steep phase? In the steep phase, the oxygen saturation of hemoglobin is very high. Less increase in PO2 leads to greater percentage of saturation of hemoglobin and therefore facilitates the oxygen loading. Also, change in the steep portion of the curve in reverse direction, that is from 60 to 10 mmHg, causes unloading of oxygen in the tissues. A small decrease in the PO2 in the tissue results in unloading of the large amount of oxygen to the tissues. The steep phase has the steep phase allows large quantities of oxygen to be released from hemoglobin in the tissue capillaries where a lower capillary PO2 prevails. So this is especially achieved by the shifting of the curve to the right side. That shift occurs mainly on the steep phase by increased H plus ions or by increased carbon dioxide. The plateau phase. The curve begins to plateau at about PO2 of 60 and flattens at about 70 mmHg. The increase in PO2 above 60 produces only a small increase in oxygen binding. Increase in PO2 from 60 to 100, the plateau region of the curve illustrates that oxygen saturation and content remains apparently constant over a wide alteration in the alveolar PO2. What is the significance of this plateau phase? So there are two significances. One, in a different situation at like high altitude or pulmonary diseases, in which moderate hypoxia, the decrease in PO2 from 95 to 60 mm occurs, the total amount of oxygen carried by hemoglobin decreases only by 5 to 10%, since HB saturation is about 90% at uh, PO2 of 60 mmHg. So thus plateau in the curve provides a safety factor, though which even a significant decrease in the lung function can allow normal saturation of hemoglobin. Second, oxygen saturation and content remain fairly constant in spite of the wide fluctuation of the alveolar PO2. For example, if PO2 rises from 100 to 120 mmHg, the hemoglobin saturation increases by only slightly, that is 97 to 98%. This is the reason why oxygen content cannot be raised appreciably by hyperventilation or breathing 100% oxygen, because HB is already completely saturated with oxygen at PO2 of 100 mmHg. This is true only for normal people at sea level. If a person has a low arterial PO2 due to the lung disease or for his ascension to the high altitude, hypoventilation or breathing 100% oxygen increases the hemoglobin saturation with oxygen as they have more deoxyhemoglobin initially. What is P50? The P50 is the level of PO2 at which 50% of the hemoglobin is saturated with hemoglobin. P50 assesses the binding affinity of hemoglobin for oxygen. In adult at sea levels, the normal P50 occurs, I mean, at partial pressure of oxygen of 27 mm of Hg. Alteration in P50 value has a greater impact on the steep phase of the curve. If P50 is high, it signifies the decrease in the affinity of hemoglobin for oxygen, which is seen in a right shift of oxygen hemoglobin equilibrium curve. Conversely, if P50 is low, it signifies the shift of the curve to the left, in which the affinity of hemoglobin for oxygen is more. This oxygen dissociation curve showing the right shift and the left shift of the curve. So usually right shift means there is a decreased affinity towards oxygen which can occur when there is increased temperature, increased 2-3 dpg levels and increased H plus ions. Conversely, the left shift means there is an increased affinity of oxygen towards hemoglobin. So that can occur in decreased temperature, decreased 2-3 dpg levels, decreased H plus ions. The factors which affects the ODC curve, there are several factors which influence the affinity of hemoglobin or oxygen for oxygen. The important factors are temperature, arterial PCO2, arterial pH, and 2-3 dpg. Rise in PCO2, a fall in pH, and a rise in temperature all shifts the curve to the right side. The effect of carbon dioxide and hydrogen ions on the affinity of hemoglobin for oxygen is known as Bohr's effect. A shift of oxyhemoglobin equilibrium curve to the right is physiologically advantageous at sea level as affinity of oxygen for hemoglobin is lowered. The rightward shift enhances the unloading of oxygen for given PO2 in the tissue, and the leftward shift increases the affinity of oxygen for affinity of hemoglobin for oxygen, thereby lowering the ability to release oxygen to the tissues. A simple way to remember the functional importance of these shifts is that an exercising muscle is warm and acidic and produces large amount of carbon dioxide, all of which favor the unloading of more oxygen to metabolically active muscles. Increase in the temperature shifts the oxyhemoglobin dissociation curve to the right, and decrease in temperature shifts the curve to the left. In other words, high temperature decreases the affinity of hemoglobin for oxygen. So this helps in the release of oxygen in metabolically active tissues in which temperature is more. So this diagram shows the effect of temperature. So this is normal curve. The increase in the temperature, favors the oxygen uh, deloading, and decrease in the temperature favors the loading of oxygen. pH. Alteration in the pH shifts the oxygen dissociation curve. Uh, okay. So Christian Bohr and Niels Bohr in 1904 demonstrated that respiratory acidosis shifts the ODC curve to the right side. Since then, the decrease in oxygen affinity in acidosis is known as Bohr's effect. A decrease in pH shifts the curve to the right, and increase in the pH shifts the curve to the left. When blood passes through the capillaries, carbon dioxide enters the red cell that decreases the intracellular pH and shifts the ODC curve to the right side. It has been noted that under normal physiological conditions, binding of about zero point 0.7 mole of H plus causes Hb to release 1 mole of oxygen. Thus, when blood passes through tissue capillaries, the acidic environment facilitates the release of oxygen from hemoglobin to the tissues. So this is the effect of uh, the pH. This is the normal curve, and the acidic shifts to the right side, and the the increase in the pH is going to shift the oxygen dissociation curve to the left side. Carbon dioxide effect. During cellular metabolism, carbon dioxide is released into the circulation that increases the generation of H plus ion and decreases the pH. This shifts the curve to the right side. So this helps the release of oxygen from the hemoglobin. The shift to right in acidosis is partially due to the effect of decrease in pH and partially uh, to the direct effect of carbon dioxide. on hemoglobin. Carbon dioxide combines with unprotonated amino groups on hemoglobin to form carbamino groups. The formation of carbamino group causes negative shift in the charge on one, uh, on one amino acid side chain that changes the conformation of hemoglobin. So this results in decrease in the oxygen affinity of hemoglobin. In metabolically active tissues, metabolic vasodilatation increases the blood flow to the tissues, increasing the oxygen transport to the area. In the area already persisting local hypercapnia and acidosis shifts the curve to the right because uh, because of a decrease in the oxygen affinity for hemoglobin that facilitates the oxygen delivery to the tissue. 2,3-DPG. Red cells lack mitochondria, therefore, anaerobic glycolysis occurs in the red cells. During glycolysis, large, quantity, large quantities of 2,3-DPG, an organic phosphate compound, is produced as a metabolic intermediary. This compound is called as 2,3-DPG. So, thus 2,3-DPG level is much higher in red cells than other cells. 2,3-DPG significantly affects the affinity of hemoglobin for oxygen. In red cells, the concentration of 2,3-DPG is almost same as that of hemoglobin. In fact, 2,3-DPG binds to hemoglobin in 1:1 stoichiometry, interacting with the central cavity uh, formed by two beta chains. 2,3-DPG has about 3.5 negative charges that interact with the 8 positively charged amino acids in the central cavity. This stabilizes the interaction of hemoglobin with oxygen. Thus, binding of 2,3-DPG with hemoglobin shifts the oxygen hemoglobin dissociation curve to the right side and facilitates the release of oxygen. At tissue level, an increase in 2,3-DPG facilitates unloading of oxygen from red cell. Hypoxia stimulates glycolysis, resulting in increased production of 2,3-DPG. The red cell 2,3-DPG increases in anemia, exercise, and hypoxic conditions like high altitude and chronic lung diseases. Therefore, in these conditions, the oxygen dissociation curve shifts to the right side. Factors that affect 2,3-DPG levels in red cells. The pH acidosis inhibits the red cell glycolysis and therefore decreases the 2,3-DPG concentration. The type of hemoglobin. The gamma chains of fetal hemoglobin have a less affinity or avidity for 2,3-DPG than beta chains of adult hemoglobin. Therefore, fetal hemoglobin has a higher affinity for oxygen. So this provides an advantage to the fetus to extract oxygen from the maternal blood in the placenta. Fetal hemoglobin shifts the oxygen hemoglobin dissociation curve to the left side. Some of the hormones like growth hormone, thyroxine, and testosterone stimulates the synthesis of 2,3-DPG. At high altitude, 2,3-DPG concentration increases substantially in red cells. So this increases the availability of the oxygen to the tissues. The hypoxia increases the 2,3-DPG production. Okay. And while you store blood, the 2,3-DPG level concentration in the red blood is going to decrease. Therefore, transfusion of stored blood decreases the oxygen transport, especially when transfused into hypoxic patients. Therefore, if blood is stored in a citrate dextrose phosphate solution, this effect is less than when stored in acid citrate dextrose solution. So this is the effect of 2,3-DPG levels. Okay, so increase in the levels shifts the oxygen dissociation curve to the right side, and decrease in 2,3-DPG levels shifts the oxygen dissociation curve to the left side. Myoglobin is the hemoglobin pigment in the muscle. Though it is similar to hemoglobin in structure, myoglobin binds one molecule of oxygen per mole. It shifts the ODC curve to the left and curve becomes hyperbolic. So that loses sigmoid shape. So this helps in the picking of oxygen from the blood. Myoglobin is more in regularly exercising muscles, especially that are trained in isometric exercise. This helps the muscle to draw oxygen from myoglobin, especially when blood flow ceases for a longer duration during sustained muscle contraction. Myoglobin is also believed to facilitate transfer of oxygen from blood to mitochondria. So this is the effect of myoglobin where the oxygen dissociation curve shifts to the left side. Okay. And compared to the fetal hemoglobin, it has more affinity towards the oxygen. Effect of carbon monoxide. The binding affinity of carbon monoxide with hemoglobin is 210 times more than oxygen. Moreover, carbon monoxide interferes with oxygen transport by competing for the same binding sites on hemoglobin. Carbon monoxide binds to hemoglobin to form carboxyhemoglobin. When the blood is 60% saturated with carbon monoxide, the oxygen content is reduced to less than 10 ml per deciliter. As the partial pressure of carbon monoxide approaches 1 mm of Hg, the Hp is fully saturated with the carbon monoxide. However, arterial PO2 may be normal as oxygen diffusion gradient remains normal. But oxygen content is greatly reduced as it can't bind to hemoglobin. So this grossly decreases the oxygen carrying capacity. Moreover, the carbon monoxide shifts the oxygen dissociation curve uh, to the left side, which decreases the oxygen release to the tissues. Therefore, severe tissue hypoxia occurs in carbon monoxide poisoning, which is fatal if not treated immediately. Normally, in healthy individuals, the carbon monoxide occupies 1 to 2% of the hemoglobin binding sites. However, in chronic cigarette smokers, traffic personnel, and in residents of crowded traffic areas, carbon monoxide concentration increases in plasma and carbon monoxide occupies about 10% of the hemoglobin. So these are the some of the factors which shifts the uh, curve to the right side, increase in the temperature, decrease in the pH, increase in PCO2, increase in 2,3-DPG, hypoxia, uh, which eventually shifts the curve to the right, increases P50, which facilitates the oxygen release. These are the factors which are going to shift the curve to the left side, that are decreased temperature, increased pH, decreased PCO2, and decreased 2,3-DPG, fetal hemoglobin, carbon monoxide. Okay, so which are going to decrease P50, that facilitates the oxygen uptake and prevents oxygen release. Oxygen delivery to the tissues. When arterial blood reaches the peripheral tissues, diffusion of oxygen occurs from the peripheral capillary blood into the tissue fluid. So this occurs along the concentration gradient of oxygen from the area of higher level to the lower level. The PO2 in systemic capillary blood is 95 mmHg, whereas PO2 in interstitial fluid that surrounds the tissue is about 40 mmHg. Thus, there is a great pressure gradient due to the considerably large pressure difference of O2 between systemic capillary blood and the tissue fluid that causes oxygen to diffuse rapidly from the capillary blood into the interstitium. Therefore, PO2 falls rapidly in the capillary to about 40 mm of Hg. Thus, the PO2 of the blood leaving tissue capillaries at the venous end of capillary and entering the systemic veins is around 40 mm of Hg. From the tissue fluid, oxygen rapidly enters in the cell. Uh, in the intracellular fluid, the PO2 is about 23 mmHg. The range is between 7 to 40 as oxygen is constantly utilized by the cell. Especially in metabolically active tissues, the gradient is more as mitochondrial oxygen utilization is more. Diffusion of oxygen to tissues by depend on two important factors, that is the rate of blood flow to the tissue and the rate of tissue metabolism. Oxygen delivery is more if the blood flow is more and the metabolic activity is more. So this diagram shows the delivery of oxygen to the capillary blood. Okay, so where PO2 is 95 and the oxygen is is going to diffuse from higher concentration to the lower concentration, and by the time it reaches the venous blood, the partial pressure of oxygen reduces to 40 mm of Hg. Bohr's effect. Any factor that shifts the oxygen hemoglobin dissociation curve to the right, uh, which is going to decrease the affinity of oxygen for hemoglobin. At tissue level, the carbon dioxide enters the blood and shifts the curve to the right. So, this helps in unloading of oxygen from hemoglobin and facilitates tissue oxygenation. So, this phenomenon is called as Bohr's effect, as it was initially described by Christian Bohr, uh, who has showed experimentally the influence of carbon dioxide tension on blood binding of oxygen. This occurs mainly due to the decreased pH in the tissue, increased temperature of the blood in the tissue also contributes. In tissue, acidification of the blood increases the affinity of oxygen to hemoglobin as deoxygenated Hb binds H plus more actively than that of oxyhemoglobin. The PCO2 level in the tissue rises, the curve shifts to the right and P50 rises. Thus, Bohr's effect helps in the unloading of oxygen and loading of carbon dioxide. Importance of oxygen saturation and content. The oxygen saturation is the ratio of amount of oxygen bound to the hemoglobin to the maximum amount of oxygen that can bind hemoglobin, that is 100% capacity. So at 100% oxygen capacity, the heme group of hemoglobin fully saturated with oxygen. Oxygen content of a blood is the volume of oxygen contained in the unit volume of blood, which includes the oxygen bound to hemoglobin and also dissolved in plasma. As the dissolved oxygen is negligible, the oxygen content depends on the concentration of hemoglobin and oxygen binding capacity of hemoglobin. The oxygen extraction is the amount of oxygen taken up by the tissues from the blood. This is an index of oxygen consumption of the tissue. It is better quantified in terms of oxygen extraction ratio, that's OER. OER is also called as oxygen coefficient ratio. That is the amount of the oxygen extracted by the tissue divided by the amount of oxygen delivered. In metabolically more active tissues like cardiac muscle, OER is high as 85% at rest. Some of the applied aspects, that is how the oxygen saturation is measured. It is measured by pulse oximetry. Pulse oximetry is a non-invasive method of measurement of oxygen saturation of hemoglobin. Oxygen saturation is continuously measured in hospitalized patients, especially patients in intensive care, by this method. Instrument used is pulse oximeter. The probe of the oximeter is attached usually to the fingertip or ear lobule, where the pulsating blood vessels are accessible externally. Red and infrared light are transmitted through the vascular bed, and pulsatile, non-pulsatile, and total absorbance are calculated. The pulsatile component of absorbance represents the arterial oxygenated blood, and the non-pulsatile component represents deoxygenated capillary and venous blood. Abnormalities of tissue oxygenation, that is less oxygenation. Less oxygenation of the tissue could be due to the less oxygen in the atmosphere, decreasing the oxygen content, uh, and decrease hemoglobin level in the blood, decreasing oxygen saturation, or decrease capacity of tissue to utilize oxygen, resulting in decreased oxygen extraction. Excess oxygenation and metabolism, uh, so which can occur, and though the tissue oxygenation is essential for life, excess of inappropriate oxygenation and oxygen metabolism, it is harmful for the tissues. During synthesis of ATP, molecular oxygen is reduced to a form of water in mitochondria. The reduction of oxygen is accomplished by addition of 4 electrons by mitochondrial electron transport system. However, leak in the electron transfer system allows oxygen to accept less than 4 electrons, so that form free radicals. Free radicals cause damage to the tissues. Lung is frequently damaged by these free radicals. Pulmonary capillaries are mainly damaged, that results in pulmonary edema. So, reactive oxygen species and antioxidants. A free radical is an atom or a molecule with an unpaired electron in its outermost orbit. Superoxide radical or hydroxyl radical are commonly produced free radicals in the body. Hydrogen peroxide can also generate hydroxyl radical. Superoxide ion reacts with nitric oxide to form peroxynitrite, which is also a free radical. These free radicals are combinedly known as reactive oxygen species (ROS). ROS cause tissue damage and promote tissue degeneration. They are sometimes called as pro-oxidants, as they are produced during this process of tissue oxidation. However, these are antioxidants in the body that prevent body from oxidative damage. Antioxidants are mainly enzymes such as superoxide dismutase, catalase, and peroxidases that neutralize ROS. The imbalance between the ROS and antioxidants, by either more production of ROS or decreased formation of proactive enzymes, results in oxidative stress. ROS are also produced during inflammations, which occurs mainly due to the respiratory burst of neutrophils. Reperfusion injury deteriorates the condition by generating local oxidative stress. So, this is in brief about the oxygen transport. Thank you.